guys, welcome to Inspiring Minds Coaching. And now we are going to watch the second. You are going to watch the second video, that is classification of the microorganisms. Here, classification I have differentiated in two form on the basis of the respiration. You can say that oxygen availability, and another one is temperature. There is a lots of classification based on other factors which I'm not including. This means they are not habitually asked in the question in the examinations. And also when you study about the pathogenicity and pathogen activity, then later you will have their specific diversity. But mostly they covered only under these two type of classifications. If I can say the 90% weightage from the on the basis of the oxygen availability. Okay, that's why it's very important to know. And directly question is asked in the examination, like facultative anaerobe, facultative aerobe. I'm going to give you the trick how to solve them, and also you will be able to discriminate them. The next section I will start with the general systemic bacteriology, because here after this video general bacteriology will be finished. Okay, then we will go to the specific one. So let's begin. Classification of microorganisms or microbes. Classification means you are going to subdivide microorganisms, because whenever you are going to decide something and divide something, then you should have a proper benchmark on the behalf of which you able to discriminate that yes, that is that kind of microorganisms, and this is that kind of microorganisms. Simply here the first one is based on the oxygen requirement. Oxygen, from the very beginning of the life, when we start studying the science, we all know that oxygen is very important for us, even for the human beings. There are two levels: at the broad level oxygen utilization and broad level oxygen system we study, and at the cellular level. And we give the term respiration, and this also respiration, and somehow it is known as breathing. Okay, here lungs involved and here proper cell involved. So technically all these things are different, but by terminology you can say they are same, but there is always a specificity while dealing with the microbes, like obligate aerobes. Simple aerobic and anaerobic, we all know that in the presence of oxygen. I'm not writing these definitions because I hope that you all guys know this. Obligate means there is oxygen requirement, and in the absence of oxygen they are unable to grow, or they are unable to show any kind of living property. Okay, the very famous example is Mycobacterium tuberculosis and Pseudomonas. Guys, in my bonus round of second section, I will give you one name nomenclature like Mycobacterium tuberculosis, and where we just use that name, like in Obligate Aerobes, from the staining process in Zell-Nelson, and then we study it later in the culture media, and now we are studying in the Obligate Aerobes. So for you it is very important to be in one line. I will give it again, like in previous lot I give. So there is Obligate Aerobes, they required oxygen. Okay, on the behalf of oxygen availability, whenever oxygen is present, only then these organisms can grow. Okay, so the second one is obligate anaerobe, means it is important for them their oxygen should not be present. What does it mean? What happens when oxygen present? When oxygen present at the cellular level, when you study in the cellular pathology, they cause a toxic effect to these microorganisms, and so they cannot grow. Similarly, if there is no requirement of no availability of the oxygen, the obligate aerobes will die, and if there is excess amount of oxygen or oxygen presents, then obligate anaerobe will die. So for you can just take them both of them as a differential, because if oxygen present, definitely these ABC sections will not be there, and if oxygen absent, you can say oxygen availability, then they are the. When oxygen absent, that is practically very easy for you to identify. Yes, that can be actinomycetes, bacteroids, or the Clostridium species. pyrifungis, Tafficella, and other disease. But whenever it says that oxygen present, so it is very complicated because the remaining others, they have somehow role of the oxygen. Okay, so for exam point of view, you can remember these things very easily. Whenever you see this A, B, C, never hesitate to answer as obligate anaerobe whenever you see actinomycetes, bacteroids, and clostridium. In obligate anaerobe, the first one actinomycetes, they are the gram-positive bacteria, and the second one bacteroids, they are the gram-negative bacteria, bacillus shape, and they cause disease to our gastrointestinal system, and that is also known as enterotoxic bacteria bacillus fragile. They cause problem with our gastrointestinal tract. Clostridium pyrifungis, Clostridium botulism, Clostridium difficile, they all are, there is a lots of variety, I mean few varieties of the Clostridium, but somehow if I say, then you can smartly see that they deal with the intestine or pelvic zone. Okay, so remember, whenever in the question on the behalf of clinical symptoms, question is asking about there is a bacterial infection to the stomach or inside the intestine, so most probably answer will be from these three if it says obligate anaerobe. Okay, keep in mind while solving question, even in the next while question session, I will again discuss these things. And another one, facultative anaerobe. Facultative means they are able to grow when oxygen present, and they are most of the microorganisms. Most. That's why I'm not writing, because they will be covered in systemic bacteriology, and also some of them is still under discovery, under lab process, so we can't fix their boundary. And the fourth one is capnophils. Capnophils, they have a specific property with the oxygen availability. Later, while I will teach you about the brucellosis and pneumococcus infection in the lungs, and also in the blood level infection, then again and again I will cover their property. For that time, I'm keeping it here as important in future, so I'm not making you confused. Let it be just capnophils. The last one, microaerophils. Very small level microaerophils, small level of oxygen is able to grow them. The first one is Campylobacter, and another one Helicobacter. They cause infection and they are very very toxic sometimes. That Helicobacter. In the croc questions, these things cover Helicobacter. Okay. And now the second classification is based on the temperature. Remember my previous video, I told you about the culture media and their differentiation on the behalf of when we change temperature, Vibrio cholera. Remember, change in pH, Vibrio cholera, and then I said that change in temperature, I give the example of Neisseria. Okay. Similarly here, based on temperature, in the staining process, I told you about sporulation and spores form in the bacteria or the microorganisms, and especially in the gram-positive bacteria. So they are very important and very infectious for the human being, also for the animal. So based on temperature, there is three types of microorganisms. The first one, psychrophils, second one, mesophils, and the third one, thermophils. Okay. For you, just you need to know like PMT examination, PMT, P first, M second, and T last. So the least temperature, or you can say the cool bacteria, that is psychophils. I personally remember it as a cool. And mesophils, most of the bacteria, they can grow in the medium temperature. And the last one, thermophils, they have very least bacteria, specific types of less bacteria. In amount, if you see, so mostly mesophils then psychrophiles, and then thermophils on the basis of amount. But when we talk about the temperature, the highest temperature is having thermophils. This graph have been asked 2 times in the USMLE examinations. They do not ask the exam. They do not ask about which one is psychrophils and which one is mesophils, which one is thermophils. They create a case, and on the behalf of clinical symptoms, their answer was a mycobacterium infection, and they just ask students to choose which category that organisms lie. So for you it is important to know that the microbial property and their ecological features, where they can grow, and even what they cause and how they cause. One step will make you able to just not only crack the examination, but also in the time of the clinical period or in your rotations, you can diagnose the particular disease in the least time, right? So this graph what says? It is all about the microorganisms, and here is temperature. Simply you can understand the first line, that pink one, is psychrophils. They are plus minus 20 degrees, means minus 20 degrees Celsius start from the plus 20 degrees Celsius. As our room temp, I will just mention here, our room temperature around 25 degrees Celsius, our body temperature around 37 degrees Celsius. And when we talk about the water fridge, 0 degrees Celsius, and when water boil, that is 100 degrees Celsius. Keep it in mind. These are basic things. I think you all guys know, but to those who don't know, just keep in mind. Okay. So here, once more in the last section, I would like to make it clear and also effective for you to learn. Just be in that form that classification of the microorganisms is based on, like a classification of microorganisms is based on two kinds of, basically they are divided on two factors according to the oxygen availability, and second one according to the temperature. According to the oxygen availability, there are 5 categories. The first one, obligate aerobes, second one, obligate anaerobe, and the third one, that here the capnophils, capnophils, and fourth one, facultative anaerobe, and the fifth one, microaerophils. Very important, facultative anaerobe, but they have lots of examples. You will learn later, step by step. Obligate anaerobe, 2 examples need to remember: TB causing, Mycobacterium tuberculosis, and Pseudomonas. MTB and Pseudomonas, they are in obligate aerobe, and obligate anaerobe, they have A, B, C, actinomycetes, bacteroids, and the last one, C, Clostridium family. Clostridium family and bacteroids, they are specifically act over the colon, human intestinal system. Mycobacterium, somehow they can infect our lungs also they can infect our other systemic organs. And in the capnophils brucellosis, that is another term, that is another complete section I will discuss there. And the microaerophils, capnophils and Helicobacter, this will be again covered in the next session, and I will tell the pathogenesis and their specific property. They form spores or they do not form, they have capsule or not, all these things will be there. And the third one, the second one, based on temperature, they have PMT, the psychrophils, mesophils, thermophils. Psychrophils, least temperature plus minus 20 degrees Celsius, mesophils, most of the microorganisms, and thermophils, least of the microorganisms. At high temperature, they grow. Our body temperature is having 37 degrees Celsius, and room temperature 25 degrees Celsius, ice 0 degrees Celsius, and boiling of the water as 100 degrees Celsius. These things keep in mind. And next, whenever I will talk about the particular systemic bacteriology, and there I will discuss the proper disease, I will take one species as a family, or a species proper, like maybe it will be Vibrio cholera, and it will be Tularemia, it will be brucellosis, it will be shigellosis. So there what I will cover, I will just use these terms. Okay. So keep in mind, I have given example and a rough idea for that. And I will say that their morphological feature, in because in systemic bacteriology, before that I will tell the structure of the microorganisms, that bacteria. And in later systemic bacteriology, I will just use the morphological term, like they have spores, like spores forming, capsulated, bacillus, spiral or coccus. And then I will go to the stain. I covered the stain in the very first in the microbiology, like I will say that this shows acid fast stain, or this one do not show acid fast stain. Then I will approach toward the pathogenesis, how they cause these kinds of disease, and then I will go for the treatment, and also there will be the etiology, why this infection have been caused that person, and what is the probability of causing that infection. Actually, this is not my term to be precise. These are the technical way to study according to the Kaplan and USMLE pattern. So, but I think when I study, for me it is difficult to know, because some basic introductory part I didn't know. So I always go to the backward and learn this here, that one is acid fast, that one is McCon के agar. So for you I am making it clear. You watch my starting 10 videos and you will be able to understand all Kaplan or USMLE notes one by one, because your concepts will be clear from the very beginning. Hope you guys like this video. Like, share and subscribe. In the next section we'll start from the question, and I will make you confident with growth medium questions, staining medium question, and also I will cover somehow gram stain. So see you in the next video with the question session. Thank you.